So in chapter 23, we talked about population genetics and microevolution, essentially. So change in allele frequency in a population over time. Right? We talked about the Grants studying Darwin's finches and how their beaks got thicker in response to a drought so they could crack tougher, older, larger seeds, essentially. But now we're going to move on and talk about macroevolution. And it's essentially the same thing, and it follows the same rules, but it's taking a step back and seeing how those same mechanisms drive not just shifting a population this way or that way, but actually diverging a population or two separate populations into multiple species, right? And we call this speciation, the origin of new species. Now, if we want to describe how one species diverges and becomes multiple species, we need to discuss a little bit about what a species is, because it's not so cut and dry, and there are a few different species concepts that we will talk about. And the first, and the one we're going to focus mostly on, is the biological species concept, since we are biologists. And this emphasizes reproductive isolation. The biological species concept states that a species is a group of individuals or populations whose members can interbreed with one another and produce viable, fertile offspring. So if you cannot mate with one another for one reason or another, that means you are in separate gene pools and therefore separate species. And whether or not two different populations or groups of organisms are really different species is not always easy to tell just by looking at them. Sometimes different species have wildly different morphology, but sometimes they don't, right? So within a species, there's a lot of diversity, right? But sometimes between two different species, just by looking, there aren't very many physical differences. And yet, if these two groups of individuals can't mate with one another and exchange DNA, then they are two different species. So when we're talking about reproductive isolation, we're talking about barriers to gene flow or barriers to two different groups producing viable, fertile offspring. Now, if they do produce offspring, these are called hybrids. And this does happen sometimes because there are some gray areas here. But sometimes these hybrids, for instance, are not viable or are not fertile or are less viable and fertile than non-hybrids. We're going to discuss several types of reproductive barriers, but they fall into two main groups, prezygotic and postzygotic. And prezygotic barriers are before fertilization, right? Before the sperm and the egg come together and produce a zygote. Anything after that is called a postzygotic barrier. Our prezygotic barriers include things like habitat isolation. So this means that two species live in different places or separated by some sort of habitat barrier, and they don't encounter one another. And if they don't encounter one another, then they're not going to be able to share genes. And this doesn't mean you have to be on opposite ends of the Grand Canyon or live thousands of miles away from each other. This could even mean that one species lives, in the tops of trees and another species lives at the base of trees or underground or something, and they're not going to run into each other very much to exchange genes. Or maybe you live in the same place, but you're active at different times of the day, right? Or different times of the year. Or if you're a plant, maybe you flower 2 weeks earlier than this other plant, even though you live in the same area. So your habitats are not isolated, but you are temporally isolated, meaning it has to do with time. You could be an animal who comes to sexual maturity in May, and another group that lives next to you comes to sexual maturity in August, right? And for that reason, you just can't mate and exchange genes. Some species are behaviorally isolated from one another. So the lab that I worked at in graduate school did a lot of research on a wolf spider called Schizocosa ocreata and another one called Schizocosa rovneri. And my old boss, George Utz, came out with kind of a seminal paper a few decades ago about behavioral isolation using these two species. So they look almost identical. The females are really, you just can't tell them apart. But the males, Ochroida, have these tufts on their legs because they do more of a visual mating display. And they do this visual mating display, whereas Rovneri mostly just has a like a vibratory mating display where they like whack the ground and show how big they are. And males of either species, right, when they see a female of either species, they can start doing their dance, but females are only receptive to males of the same species. And they did an experiment where they essentially, using carbon dioxide, you can kind of temporarily knock out arthropods, and they would knock out females of one species or the other, and males were able to successfully mate with the females when she didn't put up resistance. That's important in spiders because females occasionally will eat male spiders if they do not want to participate. But those females were then able to produce offspring. But the offspring had a mix of genes for the mating behavior, and they did a mating dance that wasn't attractive to Ochroida and that wasn't attractive to Rovneri, and so they were essentially not able to find mates themselves. All right, let's say you live in the same habitat and you operate at the same time. And even behaviorally, you're able to attract a mate. But once you get together, the lock and the key do not fit, right? This is called mechanical isolation. And one of the fastest morphological traits to evolve in insects especially are the genitalia. And so very often, the only way that you can tell two closely related species apart from one another are to flip them over and put them under a microscope and look at their genitalia. Otherwise, it's kind of impossible to tell. So if you physically, because of your bodies, cannot get together and mate, that's called mechanical isolation. Another example of this is a snail. There are two species of snail, and it's one single gene that separates them. One has a left-handed spiraled shell, the other one has a right-handed spiraled shell, but because of the way that they get together to mate, if you have a left-handed spiral shell, you can't mate with a right-handed spiral shell individual. So that single gene causes them to be different species. The final mode of reproductive isolation that is prezygotic is called gamete isolation. So let's say you live in the same habitat and are sexually reproductive at the same time, and behaviorally you're able to mate, and physically, mechanically, you're able to mate. But sometimes the sperm gets to the egg, and they just don't have the right proteins to bind together. And so the sperm can't inseminate the egg, or if they do, you have different numbers of chromosomes, and so they can't come together to form a zygote that can then divide. Whatever the reason, fertilization isn't able to happen even if the actual sexual act, copulation, is able to happen. Now, let's say you're able to have a sperm fertilize an egg. Now, there are still some post-zygotic barriers to reproduction together. So first, you may have a hybrid that is not viable, meaning it may be born, but it may not live very long. It may not develop very well. It's just not a viable organism. Or maybe it's a viable organism, but it is sterile. So mules are horses and donkeys mated together. They're very vigorous and useful on the farm, but they are sterile. Mules cannot produce offspring, and so horses and donkeys are separate species. Sometimes you may have a hybrid that is viable and fertile, but when they continue to mate either with other hybrids or with either of the parent species, the next generation or the generation after that, they become less viable and less fertile, and that's just called hybrid breakdown. And this is another way that you see reproductive barriers, post-zygotic reproductive barriers between species. Now, the biological species concept is useful, and it's probably the species concept that we'll use the most in this class and that you will use the most going forward in your careers in biology. But there are some serious limitations to it. First of all, reproductive isolation has to do with sexual reproduction. And there are huge swaths of organisms, namely prokaryotes, that do not undergo sexual reproduction. So bacteria and archaea, 2 of the 3 domains of life, have to be dealt with a different way. They can't use the biological species concept. Also, fossils, right? We look at fossilized organisms that are extinct and we separate them into different species as best we can, but we have no idea whether they would have mated with one another, right? We can't do an experiment and find out. And so it's not as useful there. Additionally, sometimes we have actual gene flow between species that we consider different. For instance, brown bears or grizzly bears and polar bears, we consider different species. But they can mate together, and they do in the wild occasionally mate together, and they produce something called a growler bear, which is viable and is fertile. Now, it doesn't happen enough in the wild that you have lots of growler bears running around, but it does happen. And so we consider them a separate species because they generally live in different areas and they don't mate together very often, and they look very different and they act different and their ecologies are different. But, you know, there's a gray area here as to what is the same species but 2 different morphs of the same species, and what is 2 legitimately separate species. And so we use other definitions of species to complement the biological species concept. For instance, the morphological species concept is probably older than the biological species concept. It allows us to define species just by their structural features. What do they look like? Now, the advantages are this can apply to asexual species, right? This can apply to fossils. We use it a lot for fossils. The downsides are it's very subjective, right? Even though biological species concept can be a little squishy, you know, growler bears, for instance, it gives you a reasonably objective standard, whereas the morphological species concept doesn't, right? We looked at the pictures earlier of those 2 finches that look almost identical, but they're 2 separate species because they won't mate. On the other hand, dogs, we have however many breeds of dogs that look incredibly different, but they really are all the same species because they can mate with one another. 2 other species concepts that are pretty useful are the ecological species concept and the phylogenetic species concept. The ecological species concept essentially views the species in terms of its ecological niche. What does it do? How does it interact with its environment? How does it eat? Where does it live? Those sorts of things. This is one of the ways that we can take asexual species and separate them, you know, without the biological species concept. And it emphasizes the role of disruptive selection, meaning if you're selected to do this and they're selected to do that, then you're probably different species, right? Now, more and more, we're also using the phylogenetic species concept, which defines a species as the smallest group of individuals on a phylogenetic tree. And that's a little tautological, but very often we're using gene data, right, or protein data, molecular data to separate species phylogenetically. And you know, that's excellent in that it allows you to work with asexual organisms, but it's still a little bit subjective. How genetically different is genetically different enough to be a different species? Because, again, sometimes a single gene can be enough to make you biologically a different species. All right, now that we've talked about species concepts, especially the biological species concept, and the role of reproductive isolation, in the next video, we're going to move to the role of geography in terms of reproductive isolation.